In this 5-mark subjective question, they are directing us to say the relationship or cumulative effect of the ribosome, transfer RNA and messenger RNA in prokaryotic translation mechanism. For this, we are drawing the translation machinery as the CBSE curriculum pattern. In this particular ribosome, the amino acid tRNA is bringing the amino acid with specific reference to codon of the mRNA. How come the amino acid tRNA is formed? Because of the amino acid tRNA synthase enzyme. This enzyme facilitates the amino acid tRNA formation. Each one of the amino acid tRNA brings a specific amino acid with specific reference to the codon of the mRNA. This particular protein synthesis is accompanied by the addition of one amino acid to the other amino acid by the peptidyl transferase enzyme of largest subunit. This event is happening between the peptidyl tRNA and amino acid tRNA. In this way, if you are representing all this information, whatever we conveyed with specific reference to this diagram, we will be getting 5 marks. The same concept, whatever we conveyed to you, we are saying again back point by point for securing 5 marks. Amino acid are linked to a specific tRNA by amino acylation of the tRNA, which is facilitated by amino acid tRNA synthase. For this mention, you will be getting half mark. For facilitating the ribosome formation, the smaller subunit and larger subunit should get attached with each other with the help of magnesium ion support. For this mention, you will be getting another half mark. In the translation initiation mechanism, AUG codon or initiation codon is facilitating the methionine addition in the first position of the peptide segment. For this narration, you will be getting half mark. In the larger subunit, there are totally 2 sites are there. One is called as amino acid tRNA site, another is called peptidyl tRNA site, and those 2 sites are mainly responsible for accommodating amino acid tRNA and peptidyl tRNA for facilitating the amino acid addition to the other amino acid for peptide bond formation so that the peptide is synthesized. For this narration, you will be getting another half mark. Ribosome proceeds in the elongation phase with the amino acid addition to the other amino acid. By means of this step, the protein is synthesized continuously after the synthesis of peptide and polypeptide. For this narration, you will be getting another half mark. We need to say the ribosome is exactly coded with the initial codon or methionine codon, which is AUG, whereas the termination codon or stop codon is narrating UAA, UAG and UGA. These number of termination codon and start codon is deciding the starting and ending of the translation. In this way, if you are narrating, we will be satisfying the remaining half mark so that we can get 5 mark for this subjective question. In this 5 mark subjective question, they narrated about the phenotypic ratio of the F2 generation or F2 progeny. If they are doing the cross between the homozygous dominant yellow round seed and homozygous recessive green wrinkled seed. With this information, we need to say the answer for the given 2 sub-question. In the first sub-question, they told about the different law of Mendel in which way it was derived. After understanding this cross, for this we are specifying the law of dominance is indicating the dominant allele is segregating the recessive allele by a phenomenon called dominance. In this, the recessive allele always gets suppressed by the dominant allele because of the nucleotide sequence of the specific allele. For this mention, you will be getting 1 mark. In the second law, we are specifying the law of segregation or purity of gamete. Allele pair segregated during the gamete formation such that the gamete receives only one of the 2 factor and there are no blending of allele or factor. In this way, if I am mentioning another 1 mark, we are securing. And for mentioning the third law, which is nothing but law of independent assortment, when 2 pair of trait are combined in a hybrid, segregation of 1 pair of character is independent of the other pair of character. For this mention, you will be getting another 1 mark. In the second sub-question, they are directing us to say the genotype of the progeny of the given 2 hybrid, yellow and wrinkled seed, green and round seed. For this, we are specifying with the appropriate genetic information. For yellow and wrinkled, we are saying capital Y, small r, one combination, capital Y, small r, other combination. For this, you will be getting 1 mark. And for mentioning the green round seed, the genotypic information, small y, capital R, small y, capital R, small r. For this mention, you will be getting another 1 mark. In this way, we are satisfying 5 mark demand of this subjective question.